All right, so I'm going to cover part 4 and part 5 of cellular respiration, wrap up chapter 7, looking at an exam here, sometime next week. Okay, this is 2021. Okay, so this section is 7.8, 7.9, regulation of aerobic respiration and oxidation without oxygen. So how is aerobic respiration regulated? It is regulated by the amount of ATP. When you have abundant ATP, okay, these processes shut down, okay? And then when you have depleted your ATP level, they turn back on. So with plentiful ATP, both glycolysis, Krebs, and I'll talk about fatty acid breakdown, they are inhibited by ATP. So we got this negative feedback loop happening here. So with depleted ATP, that means we have high ADP levels, and that activates the enzymes and the pathways to turn on and to start the process so they can make ATP. Now, there are two key points of control in glucose catabolism, okay, breaking down glucose. And the first one is phosphofructokinase, which is that enzyme found in glycolysis, if I'm not mistaken. It's the enzyme that converts fructose 6-phosphate into fructose 1,6-bisphosphate. So it slaps on a phosphate on the first carbon of fructose. So this first reaction it's not reversible. Once it's past this point, it's pretty much committed to the end. And so high levels of ATP in citrate, that's supposed to be citrate, not citrate, inhibit phosphofructokinase. The second control point that controls glucose catabolism, enzyme pyruvate dehydrogenase. Okay, so this is found in the pyruvate dehydrogenase. Yes, the Krebs cycle the link reaction where it converts pyruvate to AcetylCoA. So it's inhibited by high levels of NADH, those electron carriers. So here's glycolysis to the pyruvate oxidation. This is the link reaction to the Krebs cycle. And so these are the two key points. Phosphofructokinase, that enzyme. Okay, so how it works is ATP inhibits it. Okay, ADP activates it. The second one in these notes right here, here's pyruvate dehydrogenase. It is inhibited by the NADH. So when you have abundant NADH, like made from the Krebs cycle, it will shut off this enzyme. All right, moving on to the 9th section, oxidation without oxygen. So with oxygen, cells can produce a lot of ATP. We went through the numbers yesterday, roughly 30 to 38, kind of dependent on the organism you're looking at. We talked about leaky membranes, and then how in the process of glycolysis, that NADH, when it's being moved from glycolysis to DTC, it uses an ATP. So cells can produce a lot of ATP with oxygen. Okay, oxygen, O2 is that final electron acceptor. But what happens when you don't have any? So many prokaryotes, that's what prok's abbreviated here, use sulfur, nitrate, carbon dioxide, and some inorganic metals as their final electron acceptor in place of oxygen. So that free energy is not as great as compared to oxygen, so less ATP is produced. But the process is still respiration and not fermentation. I do want to talk about anaerobic respiration. Okay, so great examples of organisms that do anaerobic respiration, methanogens. They use carbon dioxide as their final electron acceptor. CO2 becomes methane. You find these guys in diverse environments. I believe methanogens are archaea. Okay, sulfur bacteria use sulfate. We have evidence of the first sulfur bacteria that date back to about 2.7 billion years ago, where we think we have found the organic material made by these organisms based on this isotopes of sulfur. So reducing inorganic sulfates to hydrogen sulfide, so going from SO4 to H2S. Okay, fermentation means that you're going to use organic compounds as electron acceptors. So no oxygen, cells cannot utilize an alternative acceptor, so they rely on glycolysis to produce their ATP. But remember, glycolysis only makes 2 ATP each time it goes through the process. So these electrons that are generated by glycolysis are donated to organic molecules. So an example of an organic molecule would be acetic acid, butyric acid, propanoic acid, lactic acid, or an alcohol. So lactic acid should be familiar. This is what our bodies do. When oxygen is absent inside of cells, we give it to an organic molecule like lactic acid. And then we have NADH, which will be reduced into different organic molecules. So this organic molecule will get reduced, and then NADH becomes NAD+, and this NAD+ recycles back to glycolysis to allow glycolysis to continue. Okay, so glycolysis is always happening whether or not you have oxygen or not, but it needs NAD+, okay, in its process. So aerobic respiration, oxygen required, yes. We can do substrate and oxidative. So oxidative means that we're using ATP synthase. The final electron acceptor is oxygen. We make a whole bunch, 36 to 30, that number varies. Anaerobic respiration, no oxygen is required. We can still make ATP through substrate level and oxidative. We're just using different molecules. We can still generate a lot depending on those molecules, maybe just not as great as aerobic respiration. Fermentation means no oxygen, no oxidative phosphorylation. Okay, we're making small amounts of ATP here, and we're using endogenous organic molecules, things that are found within the organism, and just look at the number of ATP here. But the thing is, we are recycling those electron carriers back to glycolysis to allow it to continue. So with oxygen, pyruvate will go through the link reaction to the Krebs cycle. Okay, generate an electron carrier. Our final electron acceptor is oxygen. Without oxygen, there are two pathways we're going to go through here: lactic acid fermentation and ethanol or alcoholic fermentation, which usually is ethanol. Okay, so here we are allowing glycolysis to continue because we are generating NAD+, which will go back to glycolysis. So let's talk about ethanol fermentation first. Ethanol fermentation is alcoholic fermentation, and eukaryotic cells, there are a few eukaryotic cells out there that are capable of this type of fermentation. So yeast enzymes tend to remove a carbon dioxide group from pyruvate, and when they cleave off a carbon, they generate this carbon dioxide, and it's released as a gas. Yeast we tend to use in bread or beers or other type of alcoholic drinks, and it causes the bread to rise. So bread that's like spongy and soft, you can just imagine ripping open a loaf of bread. You see those tiny little air holes. That's just CO2 escaping, okay? And then it produces a 2-carbon molecule called acetaldehyde. And it's this organic molecule, acetaldehyde, that accepts the pairs of electrons from the NADH, producing NAD+ and ethanol. And then what happens with the yeast is they keep producing this ethanol, and then it becomes toxic, and then it kills off the yeast. So like when you eat the bread, the yeast are dead, okay? The other type is lactic acid fermentation. Most animals regenerate NAD+ without that decarboxylation, without cleaving off that carbon. So muscle cells inside of us, we use an enzyme called lactic dehydrogenase to transfer those electrons from NADH back to pyruvate, and then that pyruvate is converted into lactic acid and then regenerates that molecule for glycolysis to continue. So circulating blood removes excess lactate. Soreness comes from when the removal cannot keep pace, and then that soreness can interfere with function and contribute to fatigue. So anaerobic tends to take place in the cytoplasm, and then aerobic respiration takes place in the mitochondria. But there are two forms: alcoholic fermentation and lactic acid fermentation. Okay, so now I'm going to move on to part 5, another short lecture here. And we are going to look at the catabolism of proteins and fats. So we've been talking about glucose and how glucose gets completely oxidized and reduced, going through glycolysis and the Krebs cycle and then the ETC. But what about proteins and fats? Because we can eat those. How does that fit into cellular respiration? So that's what we're going to talk about right here. So how do proteins and fats get broken down, and then how do they enter the cellular respiration? So we've been focused on glucose, but other organic molecules like proteins and fats are also used as sources of energy. And so just to give you an idea, proteins, when you break them down, you get amino acids, and those amino acids can play a role in forming of AcetylCoA, which can jump into the Krebs cycle and then finally get to that oxidative phosphorylation. We have polysaccharides. Those are basically the carbs okay? So like we're talking starches here, you break it down. They can also jump in at AcetylCoA and so on and so forth. And so let's talk about this more in depth. Okay, that's just a nice broad map of where they can jump in. So catabolism of proteins, what they tend to do is they tend to remove amino groups. So remember that proteins are made up of amino acids, and amino acids are made up of like 4 parts. You got your like your N group, okay, like the amino group, that's the nitrogen part, the carboxyl group, and then I think there was an OH on one end, and the other is the R group that makes that amino acid specific. Okay, so proteins are broken down into individual amino acids, and then we remove the nitrogen group. We call that deamination, and then it goes through a series of reactions to convert the carbon chain into a molecule that can create glycolysis. And it's kind of different for each amino acid dependent on the backbone of it. So alanine, okay, is an amino acid, and once we cleave off that N group and convert it, rearrange it, we can eventually get to pyruvate. Glutamate, an amino acid, can be converted into alpha-ketoglutarate. Aspartate can be converted into oxaloacetate. So this is the glutamate amino group. We're going to cleave off that N, maybe slap in an oxygen here. We can see some charges have appeared. Or no, never mind, they're here, right here. But that, once you cleave off that amino group, hey, glutamate turns into alpha-ketoglutarate. And you may remember from the Krebs cycle, alpha-ketoglutarate turned it into, I think it was like succinyl or succinate. I don't know, one of those molecules. All right, how do fatty acids jump into the process of cellular respiration? So fatty acids tend to produce acetyl groups. So you break them down into fatty acids and glycerol, and these fatty acids get oxidized in the mitochondria. And then we have these enzymes that remove the 2 carbon acetyl groups from the ends of fatty acids until all of them are converted into acetyl groups. And you're like, oh, acetyl groups, hey, that's ringing a bell. AcetylCoA, it combines with a coenzyme during the link reaction to enter the Krebs cycle. So, yeah, cool. The acetyl group combines with coenzyme A to form AcetylCoA, and that's how fatty acids jump into process of cellular respiration. Now this process is oxygen dependent, so this is the reason why aerobic exercise burns fat and anaerobic does not. Okay, so kind of cool. So this is the diagram showing my long fatty acid tails. So let's say I have a tail of 14 carbons long. I cleave off 2, so now I'm left with 12, and that 2 carbon group is an acetyl group, and it binds with coenzyme A, and it jumps into the Krebs cycle. Now I have 12 carbons. I cleave off 2, turns into AcetylCoA, jumps into the Krebs cycle. 12 becomes 10, and I just keep cleaving it until I cleaved everything off. And they enter into that link reaction to the Krebs cycle. Okay, how much ATP does catabolism of fatty acids produce? Well, if I have a 6-carbon fatty acid, okay, I go through 2 rounds of this beta oxidation to convert it into 3 molecules of acetyl-CoA. Each round would burn 1 ATP but produce an electron carrier, 2 electron carriers in the process. So I do my little math here. 1 NADH makes 2.5 ATP. That's that average we talked about in notes yesterday. 1 FADH makes 1.5 ATP. So 8 minus 2 is 6 ATP. And then we have the oxidation of each acetyl-CoA and Krebs. Wow. All right. Here we go. So 3 NADH times 2.5, 7.5. 1 FADH times 1.5 for 1.5, 1 ATP molecule. We add it all up. 3 acetyl-CoA molecules, 3 rounds of Krebs, 30 ATP produced. 3 plus 30, 36 ATP. So fatty acids, fats, there's a lot of energy stored in those carbon-hydrogen bonds. In fact, fatty acids tend to yield 20% more ATP than the respiration of glucose. If you go gram for gram, a gram of fatty acid has twice as many kilocalories as a gram of glucose. This is why fat is tended to be used as a storage molecule when you have too much energy in the animals. And then when during periods of difficulty, low food, then fat is burned. Okay, there are a small number of key intermediates that do connect the metabolic pathways. So here in blue, I have my amino acids and where they can jump in, usually that acetyl-CoA group. With the fatty acids in red, glycerol and fatty acids make up the lipids, okay? So glycerol can jump in on that PJAL, which is, I believe, G3P. And then here's the fatty acids for the acetyl-CoA. Here's some more amino acids where they can jump inside the Krebs cycle. So acetyl-CoA has many roles. We have the metabolic breakdowns of proteins and fats and the lipids that generate the acetyl-CoA. Almost all molecules catabolized for energy are converted into acetyl-CoA. So that's kind of the big jump-in point. It's also used in anaerobic metabolism. So 2 carbons are used to build up hydrocarbon chains and fatty acids. Acetyl-CoA can be produced either by channeled into fatty acids or into ATP production. So, depending on how much you have, it can be turned into fatty acids or you can use it to make ATP. But what makes the decision depends on your levels of ATP. So if you have high ATP, then fatty acid synthesis will be turned on. If you have low ATP, then the oxidative pathway will be stimulated and you'll make ATP with it. So this is a diagram showing the fatty acid synthesis with the acetyl-CoA. So, like I said, this is the big jump-in points. And if you have high ATP levels, well, then fatty acid production will be stimulated. If you have low ATP, then it will go through the oxidative stage here. All right. I think this is the final section talking about the evolution of metabolism, a hypothesis, so that means a testable statement. Earliest life forms degraded carbon-based molecules present in the environment. We think that the first organic molecules were abiotically produced, and so that first major event to harness that chemical bond energy was probably from that abiotically produced molecule. So at an early stage, organisms stored energy in bonds of ATP that we think this has been around for a really long time. So the evolution of glycolysis also occurred very early. So let me go back. Okay, so the first major event, here it is. The origin of the ability to harness chemical energy. So that's the first major event for the evolution of metabolism. The second is glycolysis, how proteins were able to jump in and help produce energy. And we think this has evolved early since glycolysis has been retained in all living organisms. All living organisms do glycolysis. It hasn't changed in over 2 billion years. Okay, a third major event is underneath the anaerobic photosynthesis part. So anaerobic photosynthesis is a different way of making that ATP instead of just obtaining it. So they use light to pump the protons out of cells and create the proton gradient for chemiosmosis. And, we didn't have high levels of oxygen back then. So photosynthesis evolved in the absence of oxygen and works well without it. The fourth major event is the substitution of water for hydrogen sulfide in photosynthesis. And this is for areas where like oxygen just might not be present. So it generates oxygen gas rather than the free sulfur. So we're making the jump for water and production of oxygen, because in early Earth history, we probably had more H2S. Okay, so for more than 2 billion years, cyanobacteria became dominant forms of life on Earth. They're the reason why oxygen gas has accumulated in the atmosphere. Fifth major event was the nitrogen fixation. So now we can utilize nitrogen, which is going to lead into proteins and nucleic acids. Just utilizing nitrogen and making those big macromolecules. Obtaining nitrogen gas requires breaking a triple bond between nitrogen, and we can't do that on our own, but bacteria can. And so they do that, and they convert it into forms like nitrates and nitrites that we can use in the production of proteins and nucleic acids. And then finally, the sixth and the final event ties into aerobic respiration and how we utilize oxygen. There's enough of it now. And so that is the final electron acceptor. And as a result, we could create a lot of ATP. So organisms didn't have to be photosynthetic. They didn't have to have chloroplasts to make their own food. They were able to get electrons from the breakdown of those organic molecules. Oofda. Okay. So this slide, I don't remember where I was going with this slide. I think it's just showing the 3 types of cells. We have archaea, no, bacteria. These are bacteria. These are photosynthetic bacteria. And then this would be our huge giant animal cell. You know what? Just screw this slide. Sorry. I'm not sure. I don't remember where I was going with this slide. I'm sure if I clicked on this website and visited it, I would know why I stuck this slide here, but I do not remember offhand. So there we go. There's chapter 7. Okay. So the guided notes, make sure you get that done so I can grade them, so you can use them to help you prepare for your exam. Your ETC quiz is coming up here, and then we're going to do a lab on fermentation.